We need to develop new sustainable development models which support conservation of resources, healthy environment for future generation. All the best and congratulations to organizing team for organizing this faculty development program very successfully. Thank you once again.
Truly said, ma'am. If we, apart from taking care of our health, if we don't take care of our surroundings, then we don't think we have a very bright future.
Yes.
It is both an honor and privilege for me to introduce the keynote speaker for today's session, a true researcher at heart and one who has contributed immensely to the field of medicine, Dr. Chandra Malladi, Senior Scientific Officer, School of Medicine, Western Sydney University, Australia. Welcome, sir. Dr. Chandra Malladi completed his education from various institutes of excellent repute and earned his doctoral degree in medical biochemistry from Delhi University. He won a junior research fellowship and a senior research fellowship from CSIR and has also undertaken many funded projects, the biggest of which was on a systems approach to understanding molecular mechanisms in health and disease. Dr. Malladi was also a research and development scientist at Mars Diagnostics, a human diagnostic company, and contributed to the development of immunodiagnostic kits such as HIV and hepatitis B. His area of expertise include protein biochemistry, molecular biology, cell signaling, and synaptic plasticity. And he has an extensive experience in scanning and transmission electron microscopy techniques. His research involves elucidating the role of mutant proteins in neurodegenerative diseases such as Alzheimer's and Parkinson's disease, understanding the molecular mechanism of hypertension, and identification and characterization of protein biomarkers in preterm birth. These are just a few of the areas he has worked on. He has developed receptor-mediated endocytosis assays to distinguish between healthy and diseased cells. He has also developed close collaborations within Children's Medical Research Institute and other research institutes in Australia. He is involved in the teaching program for UWS Medicine and has published a total of 23 papers in peer-reviewed journals with over 1,064 citations, 1 chapter, and 52 abstracts. His H index is an impressive 14, and he has been invited as a resource person at various international research centers and meetings. Multiple postdoctoral fellows and PhD students have benefited from his guidance. He has also played a major role in conducting scientific workshops at national and international conferences. These are just a few of his contributions to the field of medicine from a very long list of accomplishments and accolades. I now request Dr. Chandra Malladi to give his keynote talk on high-resolution proteomics, identification and characterization of proteins from both pathogen and host, and enlighten us. Dr. Malladi.
Thank you very much. Can you hear me?
Yes, you're
okay, excellent. Thank you very much, Pragya. And Dr. Premlata Gupta for your kind words and for inviting me to this excellent meeting, which is Advanced Opportunities and Implicit Research Technologies in Science. And I should also say my sincere appreciation to Dr. Usha Singh Rana. In spite of her busy schedule, she tried to contact me or give a talk for this National Faculty Development Program. Thank you very much from the bottom of my heart. So before going into my talk, just I wanted to give a brief words about my feeling about what is going on in India. I want to share my feelings, my thoughts, my pain, my grief, my sympathy, my condolences and as well as my prayers with you all who suffered immense loss of life, including grandparents, fathers, mothers, sisters, brothers, sons, daughters, loved ones, neighbors, strangers, and each and every one was affected. And this pandemic which is probably known as a COVID-19 disease, has no discrimination for age, gender, social status, and as well as the professions, such as doctors, nurses, educators, and students and laborers. There is no such profession which is touched it. And every family is affected either directly or indirectly. And my tributes to all of you for facing such an unprecedented global pandemic. And I also applaud your courage, resilience in facing this particular enemy and fighting spirits. But I can tell you one thing, the day is not that far to eradicate this virus, and hope that we will beat very soon and we'll come out with the colors. Thank you very much. And I will now jump into the topic. So today I'm going to talk about, let me zoom this in before taking time. Yeah, okay. So today I'm going to talk about the high resolution proteomics, identification, characterization of post-pathogen protein interaction. The reason why I took this topic is today I'm going to talk only the general talk only, but what I wanted to say is, where I am now. Sorry, there is some kind of hiccup is coming here. Okay, I'll start my video so that at least I can. Okay, now hopefully you can hear me. Okay. So today I'm going to talk about only general talk and with respect to the research technologies that contributed to this type of pandemics. So if you remember, yes, the pandemic is known to us? Yes. For example, if you see the history in 1346 to 53, if my memory correct, the Black Death, which is a bubonic plague. Plague is one of the almost 200 million people died in that particular plague. And also very famous is the 1918, which is a Spanish flu. And also the HIV, Ebola, and recently the bird flu. And now we are actually looking for the COVID-19. This is also another pandemic, which is actually, according to the WHO at the moment, is 18 million people who are affected with this one. So from bacteria to virus, these organisms jump from animal to human, and how they survive and how best we can beat these is a challenging now. Since we have the modern technologies, now we can go ahead and tackle these situations. Now, before going to this one, how these mechanism of bacteria and viruses enter the cells. Just I'm going to the fundamentals because it is a very heterogeneous group at the moment. So I want to go for the little basics, just recall our memories. The bacteria, are pathogen, or the cells, for example, the proteins or nutrients enter into the cell by various mechanisms, ranging from the phagocytosis to pinocytosis or clathrin-mediated endocytosis. My most interested is in this area, which I extensively worked on this. So this is often known as the receptor-mediated endocytosis. So what happens is, for example, if we go for the basics, if we recall, the phagocytosis is a process where the cell binds to what it wants to engulf, and then it absorbs into that. And similarly, the pinocytosis most of the time is water droplets, which actually takes inside the living organism. So these approaches are exploited by these viruses and the bacteria. And some bacteria and some viruses also adopt a different mechanism, what we call as a receptor-mediated endocytosis. Means they are like a clathrin-coated mediated endocytosis, or the caveolin, or there is a specific other mechanisms which is taken by the different mediators like the ARF6 and Rho or the CDC42. So ultimately, these pathogens will try to enter the cell and thereby they invade our immune system. Now here are some of the examples that I wanted to highlight here. So for example, which we are seeing is this particular picture is showing as a scanning electron microscope of a mouse macrophage, and this is the phagocytosine, the two chemically altered red blood cells. And if you see carefully, the red arrows are pointing the macrophages. This is what we call as a collar, and this is came from this phagocytosis macrophage, and it is engulfing. And similarly, the next one is the phagocytosis by the simple bacteria. And note, this bacteria is actually dividing. So, for example, in the olden days, and we read about the amoeba, how it takes the same way the phagocytosis. And this is another one is called the caveolin. This is a normal electron microscope, and this is a freeze-dried electron microscope picture. And you can see these are the fibroblast cells, and these are the caveolin that is taken by these fibroblast cells. And note that there is another one in the cell, which is what we call clathrin-mediated endocytosis. These are the endocytosis by a receptor-mediated mechanism, which is the clathrin-coated, which is shown here. This is the outside of the cell, and this is the inside where the clathrin coats, and the content will be slowly taken, and it closes the membrane. And I extensively worked on this one. Of course, I can give a talk after hours and hours of this particular part. There are hundreds and hundreds of proteins involved in this region. And once that is happened and it is internalized, so the most of these is a normal way of processing, taking into the inside the cell. But some of the viruses also go through the clathrin-mediated endocytosis or the caveolin or a fusion. So not necessarily every virus will pass through the different, all the mechanisms. Some are exclusively follow these what's called the entry points. And if you look into this one, how the viruses actually enters into this particular one. So now, if you are carefully look into this, the viron is budding, this is called the rubella virus, which is actually coming from the host plasma membrane. You can see this plasma membrane and is budding from this membrane. And the next one is a pseudo-colored actually, is a Ebola virus is actually emerging from this infected cell, which is shown in the yellow color. So how this, for example, the COVID actually enters in the system. Now, of course, we are all very well known, the COVID is nothing but a disease caused by new coronavirus, what we call as SARS-CoV-2, or popularly known as coronavirus. So the respiratory tract, in fact, it actually starts from the respiratory tract. So once you breathe these droplets, so what happens is it enters into the nasal cavity. So this is divided into, let's say, in the major two areas. There's upper respiratory tract, which is consisting of a nose and pharynx and lungs. These things we know very well. And the lower part of the lung is bronchus and lungs. Now, if you look into this one carefully, the airways, the cross section of this is the airways. Now, this is the trachea. There's globulate cells which produces the mucus. And any pathogen or any foreign body that enters into these airways will be taken care of by this mucus, and the ciliated epithelium will throw the foreign object outside of the lungs. So this is the normal way. But however, this virus, what it does is it bypasses the system and then they try to invade. So this is actually cross-section of a normal mouse bronchus shown here. These are the normal epithelial cells, and this is the airway. So what happened was there are tests was conducted and how this entry takes place. So what happens is, this is the figure that is showing here is a confocal image, which is a pulmonary epithelium disassembly. This is actually disassembled by the coronavirus. This particular one, which is showing in green, are the epithelial cells. These are actually destroyed by the coronavirus, which is sitting on the red color here. So this is the approach this particular virus follows. So if you go into the molecular mechanism, what exactly happens here is, first we'll see what exactly in the electron microscope it looks like. So now here, this particular slice is represented as a tomograph, which is a 5 Armstrong thickness, and showing the polymorphic SRC, COVID-2 virons. These are one particles. You can see clearly this virus is coated with a different types of decoration, which is what we call the spike proteins. So interestingly, this is a pictorial diagram which synthesizes at least 29 proteins, known proteins, which is and out of this, more predominantly is the spike protein and the membrane protein and the nuclear capsid protein and the annular protein. So these are the major proteins that is synthesized by this virus. So why we are talking about the proteins here? What is the big deal here? So if you go back, the proteins are a bunch of amino acids. If you go again, basic biology or chemistry, there are about 20 amino acids exist, and they arrange in a specific fashion, and each amino acid has its own mass, and thereby determines the molecular weight of the protein. And then these proteins play a role. This is we already studied in our basic chemistry. So what happens so there are, as I mentioned, these are the body building blocks. And if the interesting is, just I want to add a couple of things here. If, for example, if you take an E. coli cells and 1 cubic micron volume of this particular E. coli, generally actually the average value is somewhere varies between 0.5 to 2 cubic micron, depending upon the growth rate of the condition. And this having 3 to 4 million protein per cell, such a huge number in such a tiny organism. Whereas in human, the number will be 6.13 approximately million proteins present. Again, at least third of the approximation, say 20,000 different genes will make up the human genome, are activated if you talk about specifically the brain cells or neurons. So it's very interesting numbers. And if you talk about this, recently there is a guy from, what's his name, is Grant Brown here. He's a professor from Toronto University. He's a biochemist. And he analyzed the yeast cells, and he found there are about 42 million protein molecules present in a single yeast cell. Can you imagine? And interestingly, these 42 million out of some same individual proteins present somewhere between 10,000 to 20,000 molecules, and some may present even the 10. So depending upon the function of the protein, they synthesize. So why we worry about the proteins here? Because, for example, in this particular pandemic, if you take into the context, they play a very significant role in acquiring and spreading these infections. So we need to know what exactly happening between the host and as well as the pathogen. So if you look into the close-up view, and this is again the pictorial diagram which I want to show you what exactly happens here is, as I mentioned, this is the RNA virus, and it's having a lipid bilayer, and on outside what we call as the spike proteins. And these spike proteins interact with the host cell, and thereby they get internalized. If you look this one, the interaction is happening with this particular blue protein that is shown in this particular diagram. And if you see that in the close-up view, what is happening here is this is a protein, a receptor. This is an angiotensin converting enzyme 2, which is present predominantly in each and every cell. So this particular spike protein exploits that route. We have the receptors. What is the use of receptors? The receptors will uptake the various, it's like a gated channels. So it specifically passes only the specific ions or specific molecules, specific peptides only. It is not going to allow. Of course, this is sometimes it is a energy consuming process also. So this particular virus exploits this path to enter into the cell. So what is the big deal of the angiotensin converting enzyme? So this is, if you see this particular one, so what is here, what we are seeing is the network in the lungs. These particular angiotensin converting enzyme interact directly or indirectly with almost 133 proteins. And see, the arrows that is showing here, these 3 arrows that is showing is directly interacting with these angiotensin enzymes. So the virus will exploit these proteins and the machinery and for its survival with a minimal thing. So what is angiotensin converting enzyme? What it does many things. So it uses the DNA repairing and vesicular mediated transport, or the metabolism, or the digestion and absorption, and RNA replication and cell cycle. So it is a key receptor in the cell. So this particular virus exploits. So how this is actually enters, if you see the molecular schematic structure of this particular, what you call the spike protein. So what happens is, here is the spike protein that is present in the virus is having a complex structure, which is approximately somewhere between, I must say, it is somewhere between 140 to 200 kilodalton. This is the entire one. So it is again divided into 2. One is S1 and S2. These are the domains. So the S1 domain forms 3 trimers and entangle in the form of a endodomain, which is outside, because endo means out, endodomain. And sorry, the internal, sorry, endo means internal. So, and this virus protein and this S2 domain is also at the trimer associated with the spike stalk. So this is the transmembrane. So an internal domain will help to facilitate to enter into by binding first angiotensin converting enzyme. During the entry, the peptidase domain from the angiotensin converting enzyme will cleave, and thereby the virus will get fused and they internalized. That's fine. When it is internalized, so how this replication happens in this case is how it exploits. So it takes the minimal machinery of the host and thereby it exploits those. So it is an RNA virus. So naturally it actually transcribes and forms the RNA fragments, and then the various proteins are synthesized with the help of the host machinery and as well as the ribosomes, and they form and they assemble with the help of the endoplasmic reticulum with the Golgi complexes, and once it's assembled and forms in the burden from these cells. So this is the normal route. So in other words, from many places, we can have the checks and balances. In other words, we can use the drugs, we can use the antibodies to inhibit this activity. So this is, I love this slide. The reason is, this is a beautiful representation of how this particular virus exploits the host machinery for its survival. So I'll go through this very quickly. Hopefully you understand what I'm going to say is, now, if you see this one, upon entering into the cell, what happens is the spike protein, which is a glycoprotein, undergo a folding and tracking into the ether. The red one which is showing as actually related to the virus, which is a minimal in number, and the black ones which are showing as these are the proteins that interact within the virus. In other words, the virus will exploit all these proteins for its survival, and then they bury it off. So using the minimal machinery, using the maximum amount of protein machinery of the host, this virus is surviving. That's fine. We know very well every virus and bacteria will survive. So what is the big deal? If you see into the electron microscope, just I wanted to again show it in the electron microscope, how they look is, this was actually the, I think if my memory correct, this is from the lady from Kerala. She's a doctor from Kerala, and she actually regularly shows her enthusiasm and produce some articles. So I picked up from her article, actually. These are the pictures from her article. Her name is Lily, I think, yeah. So yeah, she's from Kerala. So here what we are showing is, this is the human pluripotent cells. These are stem cells. Generally, stem cells, though they have the what's called the angiotensin converting enzyme, but they are in low in levels. So these are actually induced pluripotent cells. That's called IPSCs. So what happens is, once they infected with the SARS-CoV infection, they start entering into this one. So this is a TEM image. They infected through this angiotensin converting enzyme, and they zipped into the endoplasmic reticulum. And if that was actually zoomed here, you can see this one, all this endoplasmic reticulum, and you can see all the viruses all over the place. And then what happens is, in this one, and near the cell membrane, for example, these are the cell membrane, it is breaking out. So that is shown here also very clearly, it's actually coming off. So this is electron microscope, the evidence showing that how these cells actually, so the virus behave within the cell. So now, interesting thing is, this is the first, if my memory correct, this is the, I think it is a record that getting the vaccine out such a quickly into the to the world, from laboratory to get the vaccine to the directly to the market and then your arm, it takes at least somewhere between 10 to 14 years if we are lucky. But now, in this particular pandemic situation, so we push so hard because people are using the various technologies. We are pushing, for example, neurologists working on one angle, and biochemists are working on one side, and immunologists working on one side, and epidemiologists working, and protein chemists are working on the other side. So with the help of all these people, so we were able to push these vaccines so quickly. So how this actually works, as we know very well, the phase 1 is actually just in the lab bench. We design, and then we will see how that is works. And once that is satisfactorily giving the positive results, and then we'll jump into phase 2, looking for a small cohorts. And then this is in the laboratory environment. And from here onwards, phase 3 and phase 4 is once we get success, and we'll jump into this phase 4. So at the moment, for example, we have so many vaccines are available in the world. And if my memory correct, there are approximately, I think 15, the whole, I think if my memory to know, there are 14, around 20, I think, yeah, 44 vaccines are actually under the pipeline in various countries. So now what happens is, there are different types of vaccines are now we are developing. That is, number one is a whole virus, which is a normal one. Generally what we do is a heat inactivated or cold inactivated, and then we use that virus for the antibody production. Or there is another one is the nucleic acid, now the recently actually coming. Or another popular one is the protein subunits. So once we have a protein, identified protein, and then we use the antibodies, raise that one, and we use this in a vaccine. Or we use the virus vectors also. So there are different types of vaccines that are present in the market. I think if, for example, in the, for example, we are in Australia, so we use the Pfizer vaccine, and as well as Oxford AstraZeneca. this is the first time that we got this mRNA-based vaccine. So, I mean, the results are pretty promising here. This virus, what it does is once you inject this virus, the fragment of COVID mRNA, small fragment, into your muscle, upper arm muscle, and what it does is it instructs inside the immune cell, and then it produces the spike protein. So after that, spike protein is made, this mRNA will break down. Then immune system will kick in, and thereby it will protect it. So this is a new technology that is actually developed now, which is such a rapidly we are using at this stage. And now, as we know that mRNA virus based is, people have some kind of thoughts about thinking, okay, now is it mRNA virus will integrate into the host genome and then thereby they produce the virus? No, the most of the synthesis happens, the RNA is happening in the cytoplasm. So it will not survive. And also they have a very short lifespan. So once they synthesize the spike proteins, the immune system will come into play and the mRNA will actually break down also. So it is not going to affect any way, or it is not going to interact in any way with the host genome or DNA, for example. And in fact, it never enters into the nucleus of the cell. So that is very interesting. So it is interesting to, explore this particular one, which is the new vaccines. Now, there is another vaccine recently is actually coming from US. It's called the Johnson & Johnson vaccine, which is actually developed with the spike protein. The beauty of this particular one is it stays at the room temperature and also at 4 degrees centigrade, and it only required one vaccine shot only, and it works well. But still is people are not, because the production is going very slowly. So once we get this one, this is pretty good vaccine. And of course, I think in India, we are having the Covishield and Covax. And these are actually coming from the spike protein, and Covishield is nothing but the AstraZeneca. And this is actually raised against the adenovirus, which is a common cold virus, and this will actually enhance our immune system. There is a drawbacks. They say that, most of the time people are fine, but only I think 1 in a million that happens is there is a platelets drops down, and as well as they have some kind of clots. So, but still, it actually gives almost 80 to 90% protection in these cases. And there is another one coming from the Russian one, and the Chinese, they have the one too. So in other words, what we are talking about here is these vaccines are came into the world so rapidly. This is unbelievable, actually. The people are working so hard to get this stage. And besides that, we have other treatments like the chemists. So they are actually synthesizing various types of drugs to look into this one, how best we can inhibit this viral activity within the host cell. So the popular one is remdesivir, but the problem with these drugs is they are not going to stop. They are going to only minimize the infection, and in other words, the hospitalization will be reduced, but it's not going to do other than that one. So most of these are not great at all. This is only a little bit promising, but still is not going to stop the viral spreading or viral infection. So now, as I said, besides that, now we are actually looking for the antiviral peptides. So these peptides are very short peptides. They are actually most of the time they are in the cationic one. And so what happens is these are the amphiteric peptides. They're somewhere between 29 to 42 amino acid in length. And predominantly they are on the beta sheet because they are very stable, and it's containing the 3 disulfide bonds in order to stabilize the peptides. So the mode of action is in these particular bonds are they interact with what we call the anionic phospholipids to disturb the bilayer, for example here, or it directly binds to the glycoprotein, or in other words, spike proteins, and thereby it will inhibit the entry. So again, people are looking for the antiviral peptides, but it is a long way to come. That is okay. Now, what we are talking about, that's fine. We are taking the drugs and vaccines. So why we are not able to stop this viral spread? That is very, very common question people ask. Okay, I'm taking the vaccine, so now I am now Superman. I got the immunity, so I can do whatever I like it. No, 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 no. It's not going to work that way. Because I tell you in a small example. You take a petri dish and grow the cells and infect the virus and keep doing the passages number of times. So what is happening is the virus itself is mutating and it is actually adjusting according to the particular condition. So the same way, for example, in a given country or in a given place, if we are not taking the vaccine, say for example, this is what you call the herd immunity. The herd immunity means if in the given population, if we given these vaccines, say approximately say somewhere between 80 to say 90% of the population, for example, taken this particular vaccine, the herd immunity, what we call it is. So that means the remaining rest of the 10% or 20% of the population, even they get infected, the virus may not be able to spread. In other words, there is a fight between the host and the pathogen only. That means the individual, either he will win or the pathogen will win. That's it, because it cannot escape. In other words, there is no chance for the mutation. So that was the reason the vaccines are not going to work unless we have the herd immunity. In other words, if we are vaccinated to each and every one, then there is a bright chances that we can beat this pandemic. So how we are going to see all these things, the mutations, we talk about it. So at the end of the day, we always say, okay, that's fine. So what are the technologies that we can exploit it? So for that, if I go back a little bit further, so now I'm going to this one. Oh, sorry, I forgot to this thing. So this is the crystal structure of the spike protein. This is the spike protein, which is shown in this color. And this is the angiotensin converting angiotensin converting receptor. So what is happening is these red structures there are about the mutation that happens in this particular region. So the viruses keep on mutating because it wants its own survival. So that is the way it works. So it keeps changing its mutation, and thereby it escapes the immune system, or for that matter, the vaccines, which we are talking about is. So now for that, so we need to have a better understanding of the proteins. So how this is work. Now I'm going back to the very fundamentals. I think this we learned, I think in our year 10, if my memory correct. So this is what we call the central dogma. So now what we call as, okay, from DNA to RNA, RNA to protein. So DNA transcription is transcribes to RNA, and RNA will be translated to protein. So we know this. And of course, RNA also sometimes reverse transcriptase by using the enzyme. It forms the DNA, and the DNA also do the same thing. It also transcribes also. So now, besides that, now the new technology is coming. This is called the omic field. So in this case is the relationship now is actually increased. Now, among the omic levels beyond central of this, what we call as a dogma of molecular biology, the relationship among the omics, which is shown in blue, are multifold. The enzymes that catalyzes, for example, and then central dogma are having prominent relationship. How, for example, metabolites. The metabolites forms an allosteric effects, and then sometimes they inhibit the protein, or sometimes they enhances. And the same way, the metabolite that came from the protein will also have a direct role either in the synthesis of RNA, or it is having a translational regulations. For example, most of these The regulators are nothing but the proteins only. And these proteins also have another one, what are the epigenomic interaction. This is itself is a big what's a field. I can talk hours and hours on this. So these, in other words, now our concept of this molecular dogma, which we call molecular biology, is entirely changed. Now this is, in my view, this is the right way of representation of how this exactly happened. This is nothing but based on the omic levels. These are. So now, we talk about so many things about the omics. So what is this? So generally the omics is a general terminology. We talk about the broad discipline of science and engineering for analyzing interactions of biological information. So what is OM then? OM addresses the objects of study of such fields such as genomics or interactome or the proteomics. So these are the fields where now the science is approaching. And my interest of field is, this is the area that most of the time I see it. And phosphoproteomics is one of my interesting one. And the proteomics, of course, in the lipidomics, I'm more interested. So in this one. So this is the place where now science is progressing. So why we require these things? In order to understand the molecular mechanism of these proteins when it modifies. Because, for example, if you take an example of pandemic, why I focus on this topic is on this particular one is in relation to the pandemic is how to understand the variations that are occurring within the viral surface and how that is affecting the vaccines or the drugs that we are actually designing. So we have to play ahead of game. So now there is another field of popularity is now coming called the multi-omics. So this is a really, really interesting field. So what is multi-omics? So there are now, in a layman's language, we are combining all these omics in a nutshell. So what happens in this case is first a genome, we'll go for the genome studying, and we look for the phenotypes first, and then what this phenotype is related to this, for example, in this case, the transcriptome, or in the metabolome, for example, or the microbiome. Microbiome is nothing but across the whole microbes. So we have interrelationship. So that is what we call as multi-omics. So now, if you look into this one in a layman terminology, so this is another one, the same depicted in a different form. For example, we take a tissue and we analyze the genomics. And we take the liquid biopsies and we study the proteomics. So we do the studies on the proteome, and if you take this what you call as tissues, and we study the metabolomics, and if you take the whole body and we study the radiomics. So by combining all these omics, then we can study, we can prevent, and we can provide the personalized treatments. For example, some of these things are highlighted here. So this is the field now, it's a very hot field now. So the multi-omics are the one where we can address this type of issues. So now coming back, so again, sorry, my topic is actually focused on most of the time is the current situation. So let's go back here. So as we know that when we go for checking and for whether you have a virus or not, based on your what you call that, fever and infection, etc. So, and they take the swabs here. So what they do is generally is, as you know very well, so they take the virus and they grow from here, they lyse the cells, and they go for the RT-PCR, which is a normal one. So the advantage of this one is we can track from where the virus is coming, in other words, from which individual or from what variant is coming. In other words, we can see the frame shift mutations in these genomics. But DNA is not going to tell you anything besides, yes, it is mutating. That's it. The frame shift mutation is there. Yes, a single point mutation is there. But end of the day, we need the proteins, because the proteins are the byproducts of the DNA that actually works for the interactions of the protein and the pathogen to host. So now these proteins, we digest the proteins. I'll come back in a very detailed way later stages. And then we, through a particular type of machines, and then we analyze what are the changes that are occurring in this given protein. In good olden days, if you remember, we had to purify the protein manually, and then you have to do the Edmond degradation, and then look for each amino acid changes. That takes at least a minimum 1 year to analyze it. Now we can do this thing within a day, hundreds and hundreds of proteins. That is the technology which we are talking about it is. So since this is appropriately actually fit into this what you call that, this particular national faculty development program which you guys created, and how the research is changing along with these new technologies. So that is the one which I'm talking about is. Now, when you talk about the proteome, we always associated with two-dimensional gel electrophoresis, or the electrophore separating fractionation of the proteins, and then we actually subjected them mass spectrophotometer. That's the mass spec. I'm not going to talk about the mass spec because it is itself is a huge topic. So I will give you briefly what is two-dimensional gel electrophoresis. This is a very, very powerful technique to analyze the proteins that are changing. You take the protein and you subject, because as I mentioned earlier, the proteins are nothing but a bunch of amino acids. Each amino acid has its own charge, and based on the charge, the net charge will be decided whether it's a positive or a negative. The negative charge means is a low pH, and highly positive means it is a what you call that in an alkali range. So we prepare what we call as immunoimmobilized pH gradient strips. So this is commercially available, or also we can prepare it. So these strips are containing what we call the ampholytes. These ampholytes are polycationic, polyanionic substances. These are synthetically prepared, and then we allow these to spread into this particular small strip, which is less than 0.5 millimeter thickness, these strips. And then we'll soak the complex proteins. Complex proteins are extracted, let us say, from the brain or the liver. So, and then we subject this one into the focusing, because as we know, each protein migrates according to its charge, the PI, isoelectric point. Again, go back to the very fundamental, I think if you recall your memory, the PI is the pH at which it is having the neutral charge, that protein. That is, in other words, irrespective of what are the current that you are applying, that particular protein stays there because the net charge is 0, because this is moving in pH gradient. So let us say this is the pH 7, and the protein is also pH 7. So it comes all over the place and then stops. So no matter where it is sitting, because you are applying the electric current, so naturally the positive will move the negative, negatives move the positive. So the pH 7, if it is that, and all the proteins will come and they stays there. No matter what you do, how long, how many years you apply the current, it stays there. Once you remove the electric field, then you start diffusing. So this is the advantage that we are exploiting. The charge of the protein we are exploiting to separate. The beauty of this experiment is one direction you separate on based on the isoelectric point, and another direction you separate on the molecular weight. As I mentioned, each protein is having its own molecular weight. In other words, each protein has different amino acids. So based on the amino acid numbers, it defines its weight of the protein. So generally we call the weight of the protein in terms of daltons. So we call it the kilodaltons, say 1,000 kilodaltons. 1 dalton is called 1,000 daltons. Okay, 1 kilodalton is called 1,000 daltons. So it moves in presence of, again, the electric field, and they migrate according to its charge. The beauty of this is, if you have a bunch of proteins sitting in a particular pH range, it's not necessary all proteins are same, because they migrate according to their molecular weight in terms of mobility. So this we run on the SDS electrophoresis, and then what happens is we subject it to the what you call the mass spectrophotometer. We cut the protein of interest, and then we do that analysis. The good thing is, even if it changes, even a single amino acid changes, we can see on the mass spectrophotometer. So how this was done, for example, we take the tissues or cells or whatever that is biopsies, and then we homogenize, and then we can make it as a total extract, or we can do the soluble or the cytosolic fractions and as well as the membrane fractions, and then we subject in the two-dimensional electrophoresis at right angles to the first. In other words, first one will be this direction, second one will be in the direction. So the proteins are migrates. This is actually the composite gel where you can see the blue spots are stained with the proteins are stained, and based on this molecular weight they migrate, and we will identify a protein that is of interest, and then we actually extract it and we subject it to the cryptic digestion. What does cryptic? There are a number of enzymes that we can digest it. In other words, the trypsin cleaves the lysines and arginines in that protein. If you have the lysines and arginines, it cleave and forms what we call, it disturbs the peptide bonds, and it forms what we call the peptides, a small peptides ranging from 22 to say 10 Daltons, small fragments. Then we subject it to the mass spectrophotometer, which we are going to talk about very briefly, and itself is a huge topic. And then based on its mobility and the charge, which is called the M over Z, it will migrate based on its molecular weight, the weight of that one and the charge, and they separate into the peptides. So you will see this one, and we use the database. We actually read Ignis the database. For example, human, then we use the human database. If it is a mouse, we use the mouse database, or if it is a horse, bacteria, we use that to identify the proteins. Fine. So what do we do with this? Oh, sorry. Okay, this is the one which most of the time my life sits here with these equipments. So this is the one which is we have the water equipment, so which is pretty good. So what it does is just briefly, since I saw this slide, I wanted to give you some kind of background idea what it is. This is actually the mass spectrophotomy is actually originated in the 19th century by the physicists actually. And the first known spectrophotometer was built in by, I think, the guy called J.J. Thomson, if I remember correct, in 1900s. So to study the mass over the charge.
तो फिर मैं क्या करती थी यार, the church gets a taxi भी easily.
Sorry, someone is talking. Okay, so and then so they migrate based on the positive or the negative rays. So this is the traditional way which was developed in German physics. So there are different types of mass spectrophotometers are available. So and they use this mostly the what you call the proteins and peptides we analyze this, and also the post-translational modified proteins like glycosylation, phosphorylation, acylation. So these are the ones which we look at this one. Even the protein if it is modified by an amino acid, we can clearly see, and this can be done within a day, even within 1 hour we can do this experiments. So that is the way that we rapidly, the spike proteins, why spike proteins are changing or not, immediately we can see these experiments by doing this one. So this is one of the great assets which is contributing for this pandemic, for the matter for the science, I must say this one. So this is the one which is a more advanced one compared to this one. This is if you are targeting for the single molecule, we can use it target, for example, lipid, or for example, any drug or any metabolite. It is easy to handle it, but whereas this is a much more complex one, where it gathers each and every information and can be analyzed. This is called the synapse, and this is a complex mixture only we analyze it, and it also provides a 3D structures, which is a retention, mobility, mass, and a T-wave ion mobility also. So yeah, having said, just I wanted to describe, I mean, everything is like a black box. It's very hard for us to understand what exactly going on here. So this is actually categorized, let us say, what happens inside that particular black box. So what happens is, in this case is, so this is called the LCMS. We use the liquid chromatography combined with the mass spec. That is a much better way of separation of the proteins. And what happens is we deliver the sample through this, And then what happens is, this is an atmospheric pressure at this stage, and then we use the highly pressurized chambers, and it will filter through mass to charge ratio. So once it is done, and this is the third phase, which is called it passes through the step waves. We have the different types of ion gated channels. So once they are cross these ones, because we can get a particular size of the fragment, and they enter into the quadrupole. These are made of a bunch of magnets. So they actually separate what you are looking, and then they undergo a collision, for example, and then they form what you call the, it passes through the ion passes what you call the T waves collision cells, and then they fragments, and then fragmented. Once they fragmented, they fly into what you call the time of flight. What is the time of flight? It is a high voltage we apply here orthogonally, and it accelerates the ions into this, and it reflects on the reflector tubes, and thereby it, once it reflects and it comes back to this one, and it's called the mass analyzers, which is nothing but a detector. So once we have this, we can analyze the protein, and is there any changes occurring, and if there is a changes, whatever changes are happening, and immediately we search through the database, and we know that the protein is modified or not within seconds, and thereby we can understand what is going on in the pathogenic interaction, or the any protein that you are interested, that is actually for the biomarker point of view. So recently, this particular, the COVID one I'm talking about, this one is the waters actually, they produce a kit. And this is the beauty of this kit is, it is so sensitive, even 3 femtomole, which is a 10 to the power of minus 18. We can go up to that concentration detection. Such a low concentration you can use by this equipment. No other equipment, even the radioactivity is equally comparable. So we can see. So what happens in this case is, of course, this sample preparation is antibody based. So what happens is, you have that particular, what are the digested proteins, and then we use the antibodies to this one. And then once that is done, we actually using the magnetic beads, we separate that complex, and then we actually subject it into what the peptide digestion, and we subject it to LC-MS. So what happens is, in this particular COVID one I'm talking about, this is an interesting one. That's the reason I picked up. So what happens in this particular one is the peak. This is the peak of the COVID spike protein, which is a small peptide that is generated by this digestion. And then we can quantify this COVID signature peptide at different stages of infection, So what happens is, this quantification is at the range of 3 attomole per microliter, so very, very low in concentration and very sensitive also. So the uniqueness of this particular one is, it is clean, low noise chromatography, background no noise at all, and the runtime is less than 2 minutes. That is the beauty of this particular the analyzers, or the technology, I must say. So in other words, we are reaching our goal very soon to knock out this virus. So that is the reason I wanted to emphasize here that my main talk is based on this technology, how best we utilize this. So from here onwards, I will jump into another one, which is also currently I'm working and I'm looking into this, and which is an interest of mine. And this is my babies, in other words. Okay, so now this is an intro virus, which is, of course, we are affiliated with the medical schools and the hospitals. So this intro virus, actually, what happens is, this is lives in the tummy, and then we have a vancomycin resistance bacteria. So these are the superbugs, and how best we can actually eliminate it. So I was contacted by a colleague of mine from the Liverpool hospital, and they wanted to analyze it. So they gave, hey Chandra, we have these things, what best we can do it. So I said, okay, we'll find out. So what happened, what is this one is showing is, we separated these proteins. This is a bunch of protein that is extracted from the E. coli, this particular virus, enterococcus, and then we subjected into the separation first. And then we found there are some proteins, for example, the red one is in low concentration or none or low in concentration. You can see this one, for example, these are very high concentration. The control one, which is the total soluble in control bacteria. Whereas the mutant one, it was dropped down. Or if you see carefully, these are the ones which is low, very low, or sometimes they are absent the same way. So these are the spots that we know that actually we characterize what is this is nothing but these are the various types of proteins that we found in this one. These are vancomycin resistance proteins. These are, and then these are actually the which involves in the transcriptional what's the factors during the transcription. So he was very happy, and then of course this is, I cannot talk much about it. This is actually the we are associated with some company, so, and this is a patented one, so I can't talk much about it, but certainly it is a very encouraging within a short period of time, this is, which is amazing. And similarly, this is another field that I am more interested also. So what this is showing is, this is we are looking for hypertension, the human hypertension. So it is very complex field. People die with the heart attacks and unexpectedly. I mean, there is a major killer in the world, whether it's a developing country or underdeveloped country, or wherever we go. So we wanted to understand the molecular mechanism of this, how best we can get some kind of information or the biomarkers that can be utilized for the early diagnosis. That is a goal, end goal. So we have genetically engineered mice. These are actually reflects the hypertension of the individual at different time points. For example, the 12-week-old mice exactly show the symptoms of pathological symptoms I'm talking about it is equal to the 40-year-old chap who got the hypertension, some problem with his blood pressure. So the same way 16 weeks and 20 weeks, which is equal to 16 weeks old mice is equal to approximately 55 years old, 60 years. And next is 20 years, I mean, 20 weeks is beyond that. So what we wanted to do is we want to analyze what are the differences that are happening in these particular mice, because I can't get the brain tissue from the human. It's, of course, though we have a brain bank, in Australia, it is very difficult to get the human ethics. It is extremely difficult. There are a lot of checks and balances. It's not easy. So the best option is we go for the poor mice. So then we modified, genetically modified one, and which exactly showing the human hypertension. So these were actually, we separated the proteins in a soluble form in the normotensive, means it's a normal mice, and these are hypertensive mice. So what we are seeing here is the protein species, which we are talking here is labeled as green, are the only present in normotensive compared with hypertensive. Means in hypertensive mice, these proteins were either they are down-regulated or sometimes they totally disappear. And the red, that is what I'm talking about, is totally absent. In other words, exactly the same spot. For example, if you see, and that was disappeared. And the protein species that are not related to the maturity or for that matter differentially abundant are shown in black. You can see some black spots somewhere here, you can see. So these are differentially regulated. In other words, they are unique proteins. And the transitional change between the either increase or decrease are also shown in blue colors. They are differentially 5-folds, 10-folds increase. So how best we can utilize this information? First, we took the cytosolic fractions and as well as the membrane fractions, because it is very hard to analyze in one go the whole total protein extractions, because it's too complex to understand. So we fractionate these and then we subject into the electrophorese, and then we pick up and we try to identify the proteins, and then we will see what proteins are interacting with what. This is what we call as an interactome map. So, for example, if we have this one is any protein, you've got the heat shock protein. So we find the heat shock protein, and what are the other proteins that are actually affecting with other stress proteins. So this is the way we look. If any proteins of interest, either it can be used, for example, generally we use anything that protein is specifically appeared in the infection or in the mutant mice, then we pick up and we will use as a diagnostic tool. So this is a long way to go. Of course, this is a good start, and we are doing this is using this again, the mass spectrophotometer. Now, from here onwards, if you permit me, I will take 5 minutes to jump into entirely another topic, and then I will come back for the questions. Is it okay?
Yes, sir.
Thank you. Let me turn off this, and I will go for another one. Where I am now. Okay, so show the screen, and this is the one which I wanted to talk to you. Since Pragya actually gave a very nice introduction, in that she mentioned that we are actually interested in this one. This is also another my pet project. So how the proteomics understand these preterm births? I think this is probably you might have heard. And preterm births, what is preterm births? In every year, I tell you what in a minute, in every year, we estimate approximately 15 million babies are born preterm. What is preterm? Say, in terms of this one is, normal human gestation is 280 days, which is equal to 40 days. And for the first day of the woman, the last menstrual cycle period. So, what is this preterm then? So, if the preterm, in other words, the baby matures before 40, is called as a preterm. In other words, if they deliver before 37 weeks of the pregnancy, it can be called as a preterm. Or in case of extreme situations, if the baby born between 23 weeks to 28 weeks, that's called extreme. And moderate preterm infants, they're born between 29 to 33 weeks. So we wanted to know, because what happens is preterm births are not only gives the stress and financial burden on the individuals and as well as the society and as well in terms of the governments, but also for the individual mother and baby. For example, in the long term, if the preterm baby ended up with a lot of issues, for example, difficulty in breathing and end up with diabetes and cognitive issues, and as well as the developmental problems. So how best we can actually use this particular technology to understand and as well as design some kind of biomarker for the early prediction. Coming to this, how this actually happens. So why some pregnant women actually delivers the premature baby, even the fetus is not fully developed, which is very, very tricky question to ask, because in human labor is pretty quite unique compared with any other primates, and the mechanism is not at all understand. We couldn't understand. So the preterm births actually occurs in various reasons. The most preterm births happen spontaneously, but sometimes due to early induction of labor or cesarean birth, whatever for medical or non-medical reasons. The common cause is multiple pregnancies, the infection, sometimes the chronic conditions such as diabetes and high blood pressure, which is called as the preeclampsia, or sometimes without any cause. So majorly we can say that these are, for example, multiple pregnancy, as I mentioned, preterm, premature rupture of the membranes, or idiopathic preterm labor, or preeclampsia, or second trimester bleeding not associated with the placental causes. So there are a lot of complications. Or besides that, if we go back, the racial factors. Like black women, for example, African women have twice the risk of compared with the white people, because of the some socio-economic status, medical disorders, genetic predisposition. So, or the demographic. So, for example, women with a low BMI mass or poor maternal weight gain, or the women with the age younger than 17 or 35 when they got pregnant. This is a poor education, women living alone, minimal, blah, blah, blah. There are psychological factors and post a lot of reasons. I mean, I don't want to dwell much about it. So what happens? What we need to look here is we need to concentrate. I mean, we concentrated on what we call that in the uterus. Why this spontaneous occur? Because this particular uterine wall is made up of 3 layers. One is perimetrium, second one is the myometrium, which is the middle one, and third one is endometrium. So what happens is, if the woman is not delivering prematurely, means there is something is going on in this region, because the contraction is happening, and that is the reason they are delivering the babies in an early stage. So we wanted to identify what is going on in this field. So I am more interested in the myometrial, which is mostly made up of smooth muscles. So there the contraction happens, and if contraction happens early, they ended up with the premature babies. So we wanted to identify. So the aim of this project is to understand the human labor at a molecular level, and using a top-down gel-based proteomics approach, how best we can utilize this particular technology to identify any markers that can be used. So basically, this is the same exactly, I think you might have seen the previous slide, and that is the one which is actually the tissue sample. We get it from the hospitals, and this is another perimetrium, and is endometrium. The middle one is myometrium, and we take the normal term labor because we need to compare with the controls, and as well as we have the women undergoing cesarean section because there is no option for them, so they have to undergo the cesarean section. So, and then we freeze the tissues immediately, and then we homogenize, we pulverize with this homogenizer, and then we extract both soluble and as well as the membrane proteins, and we subject it again the same way, and then we identify the proteins. So if you look at this one, these are the separation that is exactly the previously you saw. There are about 924 proteins were identified in soluble fraction. In the term level is the normal, and this is the abnormal one. There are some proteins that are present and absent, and these are present and absent in this case. The same way the membrane fractions, they are same way here also. So once we've done this one, so we cut these spots, and then we subject the mass spectrophotometer again to understand what is going on here. So we identified a bunch of proteins. So these are basically falls in the structural cytoskeleton protein. Most of the time we expected these things. These are the proteins that actually controls the contraction, relaxation, and all these associated with the muscle types. So, and also we also noticed some other what we call as a signaling molecules. And among the signaling molecules, there is a one called the beta-crystalline. Just I wanted to concentrate why it is important, because we find this one in the C-section without labor. That is very predominantly upregulated. So what is this one? This is a small heat shock protein, beta-crystalline, which is a 22 kilodalton protein. So the beauty of this protein is, if stress is induced, in other words, if the mother is stressed out, or in the hypoxia, if there is no oxygen or something like that, or the heat, oxidative stress, even the increase in the some other hormones like progesterone or inflammation, this will goes up. And what it does is actually it maintains the cell stability and survival, and protect from the cells from the harmful environment. So if you go back to the molecular level, it will actually prevent the denaturation of the aggregated proteins. And it also regulates the caspase activities. Caspase activities means they actually destroy the protein. So, and also the function is it acts as an actin polymerization, and it maintains the cytoskeleton integrity, and as well as some proteasome mediated degradation also happens for the selected proteins. So the beauty of this protein is when mothers are, let us say in this case, is stressed out for some reason, either as I mentioned, there are a lot of reasons behind that stressing, whether it's not only the physical stress, but also the mentally or the physiologically also. So what happens is the 22 kilodalton protein, they polymerizes, they aggregate, and they increase the size about 700 to 800 kilodalton. The increase in the size is nothing but induced by the stress. So 22 kilodalton protein, and when it is phosphorylated, these are the phosphorylated, it reduces the size of the oligomers. In other words, it stays as it is. So it is, everything is controlled by the lot of environmental factors and also genetic factors. So if we wanted to know if a woman is undergoing a early pregnancy, in other words, cesarean section or the preterm birth, then if we use such markers in advance so that we can predict, yes, this X and Y individual is going to go labor early. So we have mechanism to prevent those things. So that is the logic of using this particular research work. So yeah, so it is a great work. There are a lot of things we need to do. Of course, there is a lot of things we need to consider. For example, what is the ethical backgrounds and the race and the color and demography, all these things we need to include. For example, drug abusers and smokers, non-smokers. So there are a lot of factors that involve. But we got some kind of idea what exactly going to happen in these particular individuals. Of course, we find there are total of 15 proteins in these cases, and they are falling from structural to metabolic to signaling proteins. So this is, and also of course I haven't shown you is the phosphoglycoproteins. We can also identify these proteins. Since I don't have time to talk about it, so that's the reason I skipped all those things. But certainly we also looked into these phosphoproteins and glycoproteins because they regulate the mechanism also for this. And of course, this will give you some kind of understanding. And the future studies, of course, that certainly we require a lot of studies. Number one, to get that level understanding and stillbirths and preterms, and probably we can stop this premature labor if we know the answers. So yeah, so that is the work actually we are doing. And quickly, I'll take a couple of minutes to show it to you. This is actually generally way I travel from Harbour Bridge, probably the famous Harbour Bridge from Sydney. So I live in nearby, so I travel in the early morning. So the beautiful scene actually on the back is actually the ocean, and this is the hill, and the early morning sunrises. So I thought I should take a nice picture. So I wanted to share with you. And of course, I live in Western Sydney University, and just I want to add here is our university is also the top universities in the world. And there are about 11 campuses across the Sydney, and we have very versatile courses are available. And we do offer masters and as well as the PhD scholarships every year. But due to this pandemic, because of the restrictions, this year is only few scholarships were offered. And by all means, you students can go to the online survey, this Western Sydney University.edu.au, and look for the opportunities. It is a great place to work, and it's really, Aussies are, we call it Aussie. Australians are very easygoing, laid back, and very nice and funny people. So certainly you'll enjoy here. So please do apply. It is not difficult to get it, I tell you. And our PhD program is only for 3 years. It is easy to get it. Of course, you have to work hard. And in the worst scenario, in worst case, we extend for 3 months and not beyond that. So you need to submit your thesis within 3 years. In other words, it's not for 4 years or 6 years like traditional universities. So please have a look. And certainly I mentioned Pragya that when such scholarships arises, I will pass that information to her and other responsible people in your institute, so that they can pass the information, or they can put it on Blackboard, and people can have a look, and then they can apply. By all means, you are most welcome here. It's a beautiful country, to be honest. And then this is my place where I live. This is our the building. This is the School of Medicine, and this is the unique building. Of course, it stays on the big pillars, and it's a beautiful place to work. And this is the back side of the building, and this is the research lab where we actually most of the research will do. And this is the place where we have the mass spec and animal houses and other things are existing. And we have departments like pharmacology, molecular biology, and immunology, and what's the other, drug designings and everything we do here. And this wing is shared by the medical graduates. This is medical graduates, and currently actually I'm in this area where I'm talking actually. So yeah, so this is our back side of the building. So we have beautiful lawns and everything. Certainly it's a beautiful place. And this is my actually the area where generally I have to down-regulate my entire lab. So now I ended up with only this area, but that's fine. But we have plenty of space to work. So this is the place where I live and breathe and eat. I mean, sorry, I don't eat chemicals, but certainly yes. So this is the place. And the last one is, I like this one because recently actually we have a huge big moon, I think super moon we call. And that is actually, we had a eclipse at the time. So the Earth actually came in the thing and is a full lunar eclipse. So I want to share this picture, and thank you very much for your time for listening this work. Thank you very much again.
Thank you so much, sir. So there are a few questions from the participant side.
Please.
There are a few similar ones where they're talking about the comparison of vaccines. Like especially related to India, we have the two vaccines that we have, Covishield and Covaxin. Which one is better in terms of antibody production?
Okay, Covishield and both are actually raised against spike proteins. Okay, so there's not much difference, to be honest. I must say that Covishield, if I go back, I think is AstraZeneca, that is actually the Oxford. That is in, I mean, what literature says is because it keeps changing all the time. So, but the one which came from the AstraZeneca or the Oxford one is better, I mean, in my view. But end of the day, both are good in preventing the what you call it, spreading and as well as the infection. But the variant that came, I didn't talk about the mutations of these variation. The mutations that is happening in the variation in what you call the Delta virus, which is nothing but the Indian version virus, is more what you call the 18 times more infective than the one which is UK or from the South African or Brazilian. Brazilian is the next infective. And to our surprise, now in the Vietnam, I think probably you guys might have heard it, and now these the UK version and as well as the Indian version, actually they got combined and they got the hybrid virus there. In other words, all the mutations are present in that particular strain now that is coming from the Vietnam. So, but certainly not necessarily every vaccine is going to protect it. But now actually WHO is suggesting to take multi-vaccine, in other words, mixed vaccines that protects better than the single vaccine. So it is still in a very early stages, but certainly AstraZeneca or the Covax, I think that is much better than them. That is my feeling. It is my personal opinion.
Right, sir. There's another question where they're talking about the increase in immunity level by increasing the duration between two vaccines. Does it actually increase immunity level?
Okay, that's a good question. Okay, so again, the vaccines are different. For example, if you take the Johnson & Johnson vaccine, that no matter what the vaccine you are going to take it, the first vaccine when you take, first booster when you take, within 2 weeks, the IgM is the first antibody that responds. So they come within 2 weeks. So when you take the vaccine, you will be protected after 2 weeks of having the vaccine. If you take the first booster, you will be protected after 2 weeks for a short period of time. In other words, if you take the first booster, you will be protected 33% only. In other words, remaining percent, you will likely to get the infection. If you take the second booster and wait for 2 weeks to kick in the IgG, that is a predominant antibody, and once that start coming up, then your protection will raises to from 33 to 80%. Again, depends upon the vaccine to vaccine again here. If you take the mRNA vaccine, which is I'm talking about is called the Pfizer vaccine, and that will protect. Of course, you need to take the 2 boosters there, but it will protect. What we found was the variants. It is also protect in a better way compared with the other vaccines. Yes, certainly you get this one, but not necessarily you will boost the. For example, once you take the vaccine, say for example, there's no need of taking 3rd booster, but 2nd booster, there's no meaning in having another booster there because it's not going to help, because you already started developing your immunity and you raise the antibodies against that particular, what are the protein or what are the vaccine that was given to you. So yes, immunity is not going to raise, but certainly you have the memory cells whenever you come across the infection, and it will take care of that.
Right, sir. Dr. Anima Jamwal has raised her hand. I think she wants to ask a question, sir.
Yeah.
Dr. Anima Jamwal. In the meantime, sir, there's another question.
Yeah.
Where they have asked, this is regarding the preterm births that we have talked about, that do the prematurely born kids face difficulties all along their life, especially related to health?
Yes, yes, certainly. Again, depends upon if the baby is born prematurely, they have a lot of issues, because naturally they will be in the intensive care units and in the what you call the chamber, and then they have to survive for some time, and then because the development is not happened completely, so they have the cognitive problems. Certainly that happens. It's not necessary, but we have the various mechanisms to overcome that, certainly, but there is no guarantee that individual will be cured, I mean, in the long term. Yes, yes. But if you do the proper physiotherapy and if you are in the right pediatrician, yes, there is chances. Yes, certainly.
Thank you so much, sir. This informative session that you have given us. I think this was one of the most detailed presentations that we have seen, with some very beautiful images related to the COVID-19 and how SARS-CoV-2 actually attacks the cells and attaches to them. And the macrophages, the penocytosis that you had showed, all were very so beautiful. It was actually a very beautiful presentation. And the feedback that I'm getting from the participants is that they have really enjoyed your session. And it's been an informative session. Thank you so much, sir. I now request Dr. Sanjay Nagar, Head Department of Biotechnology, to say a few words.
Thank you, Dr. Chandra, for your informative and knowledgeable lecture. It is helpful to our participants. You told the recent advancement in the field of molecular biology related with bacteria and viruses is a knowledgeable lecture. Thank you, sir. Thank you.
It's my great pleasure. Actually, this is the first time that I get a chance to talk to you people, and especially Pragyaji is very kind, and she organized. I'm very, very thankful. And of course, Dr. Usha Singh Rana, and she pushed me. You have to give this talk. So I don't have any chance to escape. In other words, so thank you very much for your kind invitation, and I really enjoyed it. And I'm looking forward to talk to you. In fact, I didn't talk about another neurodegenerative disease, which is called the Charcot-Marie tooth disease. So when I get a chance, I will go in much more details of that one, because this topic, what I chosen, is a very, very easy one. But that is a hardcore topic which I'm going to talk. But again, who knows? So we will keep in touch, and then especially those students who are keen to look for the postdoc positions or PhD positions or master's position, by all means, you guys are welcome. So I'll pass it to Professor Prayagji and as well as Sanjayji, and all the details, and then thereby, I live and sleep and fight with the science. That is my passion.
Definitely, sir. We also enjoyed this session very much, and actually we look forward to hearing from you soon in a more detailed proteomic study related to the diseases that we have studied upon.
Absolutely. Thank you very much.
Now I request all those present here today to switch on your cameras for the screenshot for the photo session.
It's required.
Okay, sir. Is it done, sir?
Yes, sir. Done.
All right, sir.
Thank you very much.
As all good things come to an end, so did this wonderfully enlightening session. On behalf of ISR IPS Academy, I take this opportunity to propose a vote of thanks to those who have directly and indirectly contributed to this FDP on advanced opportunities and implicit research technologies in science, organized by our institute. At the outset, I thank our resource person, Dr. Chandra Malladi, for making us understand the proteomics of COVID-19, of preterm birth, and of hypertension so easily. We are really enlightened with your knowledge and presence. I would like to thank Architect Achal Chaudhary, President IPS Academy, and Chief Patron of this FDP, in supporting us in all our endeavors. I would like to take this opportunity to place on record our hearty thanks to our principal and patron of this FDP, Dr. Premature Gupta, for her perfect logistics support and guidance she has extended to all of us for this FDP. A special thanks to Dr. Pradeep Joshi, convener of this FDP, and to all the heads of the departments for their unflinching support and coordination. A heartfelt thanks to all the enthusiastic participants from across the nation in making this FDP a hugely successful event. Words are not sufficient to thank our organizing committee, academic committee, and technical committee, whose relentless efforts for the last many weeks have contributed to the success of this event. With these warm words, we move to the end of today's morning session. The second resume at 2 p.m. with Dr. G.M. Bhalarao from DAE UGC CSR, Kalpakkam, Tamil Nadu, as the keynote speaker. The link for the session will be shared on the group, and we have to join the meeting through Google Meet. Thank you so much.
Thank you very much for each and everyone. Thank you.
Thank you so much, sir. It was a real pleasure to listen to you.
Thank you.
And so,
sorry, go ahead.
No, sir, we had some certain more questions. We will be mailing those questions to
Absolutely,
you.
absolutely.
By today.
Thank you. Thank you very much again. See you. Bye-bye.
Good day, sir.